Okay, so talking about biochemistry. Last time we met, we talked about our chemistry lecture with the kind of beginning half of chapter 2 in your textbook, where we studied and learned about bonds and sharing electrons, transferring electrons, and everything having to do with matter. In this lecture, we're going to apply what we learned last time to biology. Biology, if you're not familiar with that, is the study of life. So biochemistry, therefore, is the study of chemicals important for living things, like the one that's animated here in our GIF, which is, I'm sure everybody recognizes, DNA. Okay, one of the most important biomolecules, one of the most complex. We'll talk about DNA towards the very end of the lecture. I'm not sure if we'll get that far today or not, but we'll see. DNA is an example of a organic compound. Okay, we're not going to talk about organics quite yet. We're going to first talk about inorganics. Inorganics and organic compounds are the two principal classes of the chemicals that we find in our body. And we'll start here talking about inorganics. Features of those inorganic compounds include the following. They're small. Looking at the examples I've included here, O2 is just 2 atoms of oxygen. HCl, hydrochloric acid, is a hydrogen atom and a chloride atom. They're actually ions, so they're bonded together. CO2 is 3, H2O is 3 atoms. So very small, especially compared to what we'll see later on this afternoon when we get to the organics. Organic compounds are built around carbon and have lots and lots of carbon. So we describe them as having what we call carbon chains. In the case of inorganics, they do not have carbon chains. Now, like in carbon dioxide, CO2, it does have 1 atom of carbon, but 1 is not a chain. Okay. They either contain ionic or covalent bonds. Remember, ionic bonds are like what happens when the hydrogen ion H+ is chemically attracted to a Cl- ion. Cations and anions are oppositely attracted or oppositely charged, so they're attracted to each other, like we talked about the other day. Or they will contain covalent bonds, which are where the electrons are shared. We cannot use inorganic compounds in our cells to perform complicated biological functions, but that doesn't mean that they're, useless or not important to us. For example, O2, we commonly, just refer to that as oxygen. That's oxygen gas. We survive a matter of minutes without it. So it's obviously very, very important to us. Okay. It's what we use to transfer. We use it in our mitochondria. So we'll kind of talk more about this next class period, probably, to make ATP, to make energy. HCl, already kind of mentioned, that's hydrochloric acid. That's a chemical that you'll find in your stomach to help you digest your proteins that you've ingested. Carbon dioxide, that's a waste product formed whenever we make ATP, and we must exhale it out. And that's what we breathe off when we blow air out of our lungs. And then H2O, water, as we commonly call it, is going to make up 55 to 60% of a lean adult's total body mass. And we've already been talking about water even back to the very first lecture when we talked about the different fluids of the body: intracellular, extracellular, interstitial. Okay? So it's a big part of us, and it also is going to provide us with a lot of unique features, or water has a lot of unique properties that are important for us. We'll talk about that more on the next slide. And there's also other salts, acids, and bases I didn't include that are important as well. Okay, water can be described as the most important and abundant inorganic compound in all living systems. So it's not just important in us. It's not just important in, animals that live in water, like, fish and dolphins and sea turtles. It's found and abundant in, plants. That's, we all know you have to water a plant for it to grow, but it's found in every living thing, whether it's as simple as bacteria or as complicated as we are. Without it, we would die in a matter of days, and that could be even less than days if you're, in the middle of a desert and it's 160 degrees outside. You're sweating like crazy, and you're not getting any water. You're going to dehydrate pretty quickly. Several properties of water make it indispensable for life. It is a chemical reactant. It serves as a medium for most chemical reactions in the body and participates as a reactant or product in certain reactions. So thinking about what we've talked about previously, we learned that water is everywhere inside our body, inside our cells, between our cells. Well, it's the medium, the place where most chemical reactions take place. We talked about the terms hydrolysis and dehydration synthesis towards the end of class last time. Hydrolysis, remember, that's where water is used to break bonds when we're performing a catabolic reaction. Dehydration synthesis, the opposite of that, is when water is removed and bonds are formed, or we build something up in an anabolic reaction. So water is involved and can be the starting or the ending materials in certain chemical reactions. Okay, water has a high heat capacity, meaning that it can absorb or release a relatively large amount of heat with only a modest change in its own temperature, which is very helpful for us because it helps maintain body temperature during environmental temperature changes. Thinking about going outside on a really cold day. Today's not such a cold day, but we'll probably be experiencing this in the next few days as the weather kind of changes. Your body temperature doesn't just plummet as soon as you walk outside, even if you were to walk outside and it's 10 degrees and you're naked. Okay, a lot of that is because of the water that is inside of us, and it basically kind of holds that heat, so it's not just going to go away. And we talked about this the other day, which leads me into the next bullet there. Water has a high heat of vaporization. It requires a large amount of heat to change water from the liquid form into a gaseous form. Okay, and I mentioned this as the explanation for why water takes a while to boil in a pot on the stove. It doesn't just instantly boil. It takes a few minutes. Okay, well, we're not a pot of boiling water, but when water evaporates from our skin, it turns from liquid to a gas. It pulls that heat along with it, and so it removes a large amount of heat in order to cool the body. And that's going to explain why dry fit, Under Armour, Nike Fit clothing is better if you're going to be exercising in a really hot environment and sweating a whole bunch than just like wearing a cotton t-shirt. Cotton t-shirts just get soaked, and the sweat doesn't evaporate from the skin like it would or does with those moisture wicking materials, and that's why it's not as efficient as cooling the body. Okay, water is a major component of mucus and other lubricating fluids in the body, so it helps with allowing for joint movement. We talked in anatomy this morning about synovial fluid in our joints. It's also found in the places like the membranes around our heart and lungs to allow organs to touch and slide over one another without friction going on, or with minimal friction anyway. Water acts as a solvent, meaning that it dissolves ionized or polar substances, what we call solutes, forming what we call solution, which relates to its polarity. We'll talk more about this on the next slide. And also, due to its polarity, water is cohesive, meaning that the water molecules hold together, and the formation of hydrogen bonds occurs. Okay? Hydrogen bonds, remember, are those what we call intermolecular bonds, where like a water is attracted to another water and attracted to another water. And we mentioned that the other day. So hopefully that rings a bell. So let's talk a little bit more about water as a solvent. One of the most important things that our water does for us, which we've been talking about since the very first lecture of the semester last week, when we introduced intracellular fluid, extracellular fluid, and we defined what a body fluid is. Okay. In a solution, a substance, what we call the solvent, dissolves another substance, what we call the solute. So in other words, in our body, for example, we have sweat, which is predominantly water, which acts as the solvent. that dissolves other stuff, and notably that would be like salts, Na and Cl ions, sodium and chloride ions. Okay. Usually there's more of the solvent than the solute in a solution. You can see like the little blue dots as they kind of are starting to dissipate here, but the blue dots will be the water molecules, a lot more of those than whatever these little guys are, which would be the solutes. And the ability of water to form solutions is very essential to our health and survival because water can dissolve so many different substances. It is an ideal medium for chemical reactions. It's where chemical reactions are taking place. We just mentioned that on the previous page. It enables dissolved reactants to collide and form products. Water also dissolves waste products, which then allows them to be flushed out of the body in the urine. And this is all going to relate to what we talked about the other day with the unequal sharing of electrons between the oxygen and the hydrogens, where oxygen has a stronger pull and thus the electrons are kind of more towards the oxygen than they are the hydrogens, which explains why the oxygen becomes slightly negatively charged, the hydrogens become slightly positively charged. And as we learned, opposites attract, right? Well, look what happens to a crystal of sodium chloride or salt if exposed to water, okay? Notice the sodium ion here becomes directly surrounded by all those negatively charged or slightly negatively charged oxygens, because plus and minus, that's opposite, they're attracted to one another, whereas the chloride ions become separated and then are surrounded by the hydrogen ions, which are slightly positive. Okay? And that explains why water dissolves things like salt and other ions, which we mentioned, we call things like sodium and chloride in our blood and in our fluids electrolytes. Okay. So hopefully you guys remember that from the other day. Okay, a mixture is a combination of elements or compounds that are physically blended together, but not bound by chemical bonds. In our body, there are three common liquid mixtures: solutions, colloids, and suspensions, although most of what we talk about are in that solutions category. Okay. Once mixed together, solutes in a solution remain evenly dispersed among the solvent molecules. Because the solute particles in a solution are very small, a solution looks clear and transparent. Thinking like salt water, salt water is clear, just like, fresh water, right? You can't have a glass of salt water and a glass of fresh water sitting on a table and be able to really tell them apart just by looking at them, okay? And that's because those sodium and those chloride ions have just dispersed and they're so small that they don't break up the light that passes through the fluid. Now, in a colloid, it's different from a solution mainly because of the size of the particles dissolved. They are large enough to scatter the light, which causes a colloid to appear a little bit more translucent or opaque, kind of cloudier. We don't really talk about colloids except one other time this semester, and that's when we talk about the thyroid gland, which actually I was talking a little bit about this morning in anatomy. The thyroid produces a couple of hormones that regulate our metabolism. And those two hormones collect inside the fluid of the thyroid called colloid. And that's really the only time we ever really mention it outside of right now. Now, unlike solutions and colloids, the solutes in a suspension are so large that eventually they'll settle out. And your blood is a good example of a suspension. Okay, looking at the bottom of the page, we can show what can happen to blood if left in a test tube, or if run through a special type of machine called a centrifuge that spins really, 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 really fast. What happens is the red blood cells, which are the most dense component in our blood, the most prominent parts of our blood, they filter down to the bottom, okay, because they're the heaviest as far as density goes. The white blood cells and the platelets would sit on top, what we call a buffy coat. And in the liquid portion of your blood, what we call plasma, which is predominantly water, but also contains things like ions and gases and waste products, will sit on top. Okay. Now, if you were to pull the plasma, the liquid, out of this test tube, then that would be a solution. So blood in a whole sense is a suspension, but it has a liquid portion, plasma, which is a solution, if that makes sense. Okay, good. Now, when an inorganic acid, a base, or a salt dissolves in water, it will dissociate. Okay, this word dissociate just means that they separate into ions and become surrounded by water molecules. We saw that here in our picture a couple of slides ago. Okay, we can see kind of the same thing taking place if I jump ahead. Acid, a base, and a salt. Hydrochloric acid, potassium hydroxide, potassium chloride. Those ionic bonds that hold the acid, the base, or the salt together dissociate. They break apart. That's what that word dissociate means. Okay. Let's talk about acids and bases, and then we'll talk a little bit about salts, but mainly here we'll be talking acids and bases. An acid is a substance that dissociates into one or more hydrogen ions, H plus, and one or more anions. Anion is like what hydroxide is, has a negative charge. We classify acids or call them, refer to them as proton donors, since hydrogen ions are basically just composed of a single proton. Okay, if you think back to the other day when we talked about atomic number, atomic mass, and that kind of stuff, we said hydrogen has an atomic number of 1, right? That means it's got 1 proton and 1 electron, okay? But now if we are dealing with an ion that's H plus, that's telling us that there's not an equal number of protons and electrons anymore, okay? Well, H plus is telling us that we've lost that 1 electron that hydrogen has, okay? So it had 1 electron, 1 proton, it's lost its electron. Most hydrogen doesn't have a neutron, so what's left is just that 1 proton. So a lot of times you'll see H plus just described as a proton, okay? Now, the level of acidity, or so how strong of an acid a solution has or is, depends on the number of hydrogen ions a particular acid will release compared to the number of hydroxide ions, which would be this OH negative, okay? And basically, you can kind of think of it. Need some paper real quick. Let me grab something. So it's as simple as comparing the number of an acid will have more hydrogen ions than hydroxides. That's a greater than symbol. So in the case of our 2 examples, if you were to dissociate H2O, we'd form 1 hydrogen ion, 1 hydroxide ion. Well, we're comparing the ratio there, it's 1 to 1. So is water an acid? Is this number more than this number? No, it's the same, right? Okay. What about HCl? Dissociates to form a hydrogen ion and a chloride ion, where now the ratio is 1 to 0. So this hydrochloric acid, obviously hence the name, is an acid. Okay. Bases, or an alkaline compound, are electrolytes that when dissociated in solution, they shift the balance of the hydrogen ions, hydroxides, in favor of hydroxides. So in other words, a base is this. More hydroxides than hydrogen ions. So our example here, sodium hydroxide dissociates to one sodium ion, one hydroxide ion. There are no hydrogen ions. There is a hydroxide, so it's a favor towards hydroxide. That means sodium hydroxide is a base. Going back to water, H2O, is water a base? It's not an acid, nor is it a base, because it's not one greater than the other. Instead, they're equal, which means that water is neutral, where the number of these ions is equal to each other. Okay, so we said that acids are proton donors. Bases are often called proton acceptors because they remove those hydrogen ions from a solution. So they accept them, they pull them away. Okay. Now, I'm not going to ask you on the exam to do any like the dissociation stuff and figure out the relationship or ratio, number of hydrogen ions or hydroxides. What I'm going to ask you to know, though, is a value on the pH scale, or know what the pH scale means and be able to read it and tell me if you're dealing with an acid, a base, or something that's neutral. pH term refers to the power of hydrogen. That's an actual abbreviation, and that's why the letter H there is capitalized, because if you are looking at the chemical formula or the abbreviation for hydrogen, it's one capital letter H. Okay. This is used to mean the relative hydrogen ion concentration of a solution. It's a very complicated logarithmic scale that you can do calculations on, but not something I'm going to ask you to know. Simplified version of it is just what's illustrated here. Going from 0 to 14, we can measure whether we have a base, acid, or something that's neutral. Right smack dab between 0 and 14 is the number 7.0. 7.0, which is where pure water is the point where the number of hydrogen ions and hydroxide ions are equal. Okay. Anything above 7.0 is a basic solution, or is a base. Okay. Anything below 7.0 is acidic. Now, the farther you get from 7, so closer to 14, the stronger the base. The farther you get towards 0, the stronger the acid. Okay. That is something I might ask you on the exam. I might say, which of the following is a stronger acid? 4.0 or 2.2? You'd have to say 2.2. Maybe I'll just ask you on the exam. Human blood has a pH of 7.4. Is that an acid, a base, or is that neutral? And you have to say 7.4 is a base. It's a really weak base, okay, but it's a base nonetheless because it's above 7.0. Okay. Now, table 2.4 lists off selected pH values of substances. Some of these we'll kind of talk about more than others. Like we will talk later on the semester about your stomach juice, which is super acidic. Okay. Really, the only one that I will directly want you to know this semester is your blood. Okay. Which I said is around 7.4, somewhere in this range, 7.35 to 7.45. And we'll talk a little bit more about that here on this page. One of the most important things to maintain as far as homeostasis goes is the pH of our body fluids. Because if your pH is off, that can really shut you down. Okay. Now, comparing the pH of one fluid in our body to another, it will be, in many cases, very different. Going back to the table on this previous page, blood is 7.35 to 7.45, whereas your gastric juice inside your stomach could be like 1.5, 1.8, 1.10, okay, or 1.9, all the way up to 3.0. Okay, whereas in your saliva inside your mouth, it's a pretty weak acid, but it's an acid nonetheless. Those are very, very different values, okay? So the pH of these body fluids can differ greatly. One thing that they all share is the normal limits for which those fluids can be at as far as pH goes is quite narrow, okay? We can't see drastic changes in pH. You know, for blood, we like to keep it within a tenth, 7.35 to 7.45, which, as we already have said, is slightly alkaline or basic. Okay. It is possible to see a drop below 7.35 and a elevation above 7.45. We call these conditions an acidosis and an alkalosis. But hopefully those are not long-term situations, because that can be detrimental to your health. Now, it's kind of misleading when you say, you know, if your blood dropped to like 7.32, is that truly your blood as an acid? No, because it's still above 7.0, but it's trending towards acidosis. It's becoming closer to 7 and, you know, closer to below it. Okay. Now, even though strong acids and bases are continually taken into and formed in our body, just look at the previous page and you'll see things like, you know, Coca-Cola having a real low pH and stuff like that. The pH of our fluids inside and outside your cell will stay relatively constant. Okay. And this is due to the presence of buffer systems. A buffer system is going to function to convert strong acids or bases into weak acids or bases. Okay. Essentially, you can think of a buffer system and buffers, which are the chemical compounds that do the buffering. It's just if you want to say, what does a buffer system do, or what does a buffer do? It just maintains pH. Okay. It's very complicated in how it all works, but they keep your pH in check. Okay. One of the most important buffer systems is the carbonic acid bicarbonate buffer system. We'll talk about that later on the semester, which acts to buffer pH changes in your blood. Okay. Now, there are two main systems that are most responsible for maintaining your body's pH. Okay. So the other day, we learned, you know, that generally, or depending on how you classify things out, you can classify the body as having 11 or 12 systems, right? Well, two of those that were in that table are most responsible for maintaining pH. Any idea which of the two they are? How about some guesses? Anybody got any guesses? Who wants to talk today? That's okay. Yesterday I taught this and I had a lot of people guessing things like the endocrine system, lymphatic system, cardiovascular system. None of those are correct. Nervous system, most responsible for maintaining homeostasis, not the system that maintains pH. Okay. This might surprise you to find out that your urinary system and your respiratory system, those are the two systems that most regulate your pH. And you might be wondering, well, why, how does my respiratory system do that? Well, breathing regulates the pH. Okay. When you exhale CO2, you are keeping your body from becoming acidic. Because if carbon dioxide builds up in your body, it will combine with water, and that forms an acid. Okay. And we can breathe really rapidly, and thus we can regulate our pH pretty quickly. It's important for short-term acid-base balance, or pH balance. Your urinary system utilizes just the act of peeing. We can get hydrogen ions out of our body that way, so we can pee out acids. We can hold back acids if our body is becoming too basic. So that's kind of cool. That's much more long-term as far as acid-base balance. So we'll talk more about that stuff towards the very end of the semester when we talk about the respiratory and urinary system. That's like the last 2 units. Okay, so that's acids and bases. Pretty close in similarity to those are salts, except when they dissolve in water, the ions that dissociate are neither going to be hydrogen ions or hydroxides. Okay, so in this case, like potassium chloride dissociates to potassium and chloride ions. Neither of those are a hydrogen ion or a hydroxide. Now, what's cool is you can take an acid and a base and they can react with one another to form a salt, like, the chloride from this, the potassium from this, and that's potassium chloride. Salts are very important in the human body to act as electrolytes, important for carrying electrical currents, especially in our nervous and muscular tissues. When we talk about neurons sending signals called action potential or nerve signal, we're going to talk about two ions at great detail, and that would be potassium and sodium. To a lesser extent, we will also talk a little bit about chloride, but sodium and potassium are the two that we talk about the most, and that's what we'll be getting into here in the next couple of weeks in Unit 2. All right, so that's the inorganic compounds important to the body, and just a little bit about them. Rest of the lecture is going to be dedicated to the organics. This is the other principal class of compounds in the body. And a key feature of organic compounds is the presence of carbon, as I was kind of already talking. And if you take a class called organic chemistry, or if you just study organic chemistry, you're going to talk at great length about carbon. Now, why is carbon such an important feature in organic compounds? Well, because of the ability of it to covalently bond up to 4 times in a variety of ways, straight chains, branch chains, rings, you have double bonds and single bonds and all sorts of different things. Now, I'm going to jump ahead a couple slides and explain why carbon is the backbone. It is the most versatile building block of our organic molecules, compounds, whatever you want to say, because they have an atomic number of 6. They will have 6 electrons. Okay, we talked about this the other day. 2 go in the first ring, which leaves 1, 2, 3, 4 in the outside ring. Now, in this current state, is carbon satisfied? Remember, the octet rule states that we want to have how many? 8. And to get from 4 to 8, we need 4 more, right? Well, if you wanted to be super simple, you could single covalent bond there, single covalent bond there, single covalent bond there, single covalent bond there. Okay. So that's an easy way to put carbon in a satisfied state. 4 covalent bonds. If you wanted to get even crazier, you could do 2 double covalent bonds. A double and 2 singles, a triple and a single. You can make branches go in all 4 different directions. You can go in 2 different directions. You can go in 1 direction. I mean, there are so many different possibilities where carbon is the central point from which the molecule can branch off into. It's not always, but into possibly 4 different directions. Carbon has covalent compatibility. I mean, it likes to share these electrons with many different elements. Remember those 4 major elements? Carbon was one of them. What were the other 3? John, what's it short for? Carbon, hydrogen, oxygen, nitrogen, right? Okay. Hydrogen, oxygen, and nitrogen are also very fond of bonding covalently, sharing electrons. Okay. And especially hydrogen and carbon get along really well. And we'll see what we call hydrocarbons a lot here today. We've got carbons and hydrogens really intertwined. Okay. Now, we can describe an organic compound as having many monomers forming what's called a polymer. Okay. Think of the bricks that are used to build a brick house. Okay. We all know by now, hopefully, what the prefix mono means, right? 1. Okay. So a monomer is like 1 brick. Poly means many. So a polymer is like the whole house itself, made up of hundreds or thousands of bricks, depending on whatever how big the structure is. Okay, well, you build a house out of bricks by starting with one brick, bonded to another, and then bonded to another. That's how we form organic compounds. That process is called dehydration synthesis. That's this illustration we saw the other day. One building block, another building block, or one brick and another brick bonded together. Water is removed, dehydration synthesis. Okay. Now, just like a brick house, you can break it down, right? Okay. Brick houses get torn down all the time. Polymers can be broken down, or in this case, a dimer can be broken down through that process, what we called the other day, hydrolysis. Okay. That's what that other figure was showing. Okay. Now, what you're going to find in these organic compounds are not just hydrogens and carbons, but instead, you'll also find attached to that carbon skeleton distinctive functional groups. Okay. Functional groups are groupings of specific atoms that confer chemical properties to the organic molecules. And looking at table 2.5 from your textbook, you can see some of the most important or major functional groups found in our organic molecules. Okay. If you look at a molecule and you see this collection of atoms, you would call that a hydroxyl. Okay. We've mentioned ATP. The other day we talked about ATP having 3 phosphates, right? That's what we call it, adenosine triphosphate. Here's what a phosphate looks like. It's this collection of atoms. ATP has 3 of these together. Okay. Amino acids, guess what they have? Either this or this, what we call an amino group. Okay. And it's these things that really make those 4 different organic compounds different from one another. It's what makes a fat different from a carbohydrate, and a protein's different than a nucleic acid. And so I've already kind of read through all this stuff at the top. I'll just read through the bottom. Covalent bonds link carbon atoms together in long chains that form the skeletal framework for organic molecules. These carbon skeletons may vary in length, shape, as some will be straight chain, branched, ring, etc. Number and location of double bonds. Other elements covalently bonded to available sites. That would be the functional groups that we just talked about. And this variation in carbon skeletons contributes to the complexity and diversity of organic molecules. So, what are the organic molecules of the body? Well, there are 4 main groups: carbs, lipids, proteins, and nucleic acids. We're going to talk about each one of these, what they do for us, and then the different forms of them. Starting with carbs, then we'll talk lipids. What are lipids most commonly called? Fats, proteins, and then the complicated one here on the far right, which is showing you DNA. The most complex are the nucleic acids. All right, let's talk carbs. What people seem to fear and hate at the same time. But are very important to us because, as you can see, they provide most of the energy needed for life. They include sugars, starches, glycogen, and cellulose. So a packet of table sugar is just a carbohydrate. It's a really simple form of carbohydrate. Now, some carbs are converted to other substances, which are used to rebuild structures, but most importantly, we use carbohydrates to generate ATP. Other carbs can function as food reserves. We can store them in our body. In case we aren't getting enough calories to survive. We classify carbs into 3 classes based on the length of their carbon chains. There's that prefix mono again, another prefix di, and then poly. A monosaccharide is like having 1 brick. A disaccharide is like 2 of them put together, and then poly would be 3, or in many cases, a heck of a lot more than just 3. We'll start here on this next slide by talking about the monos and the dis. Monosaccharides are going to be the shortest and simplest of our carbohydrates. Generally have about 3 to 7 carbons total. We classify the monos as the monomers. These are the building blocks for all other carbohydrates. Example would be glucose, the one we'll talk about the most this semester, which I've already mentioned before, is C6H12O6. If you were to go through and count up, you'd be able to find 12 hydrogens, C6H12O6, you'd be able to find 6 oxygens. Looking here, though, we don't really see very many carbons. There's one right there, but right here, I don't know why they do this, but there should be a letter C there for a carbon. There'd be another one there, and one right there. So just where the points are and there's nothing, there would be a carbon there. So that's what glucose is. Okay. If you take one of these 3 and bond it to another one, for example, glucose and fructose, you can form what's called a disaccharide, like what's called sucrose, what we commonly call our table sugar, 2 monosaccharides bonded together by way of dehydration. Synthesis, okay. Collectively, we call our monos and disaccharides the simple sugars. This is what should be minimal in a person's diet. Okay. These are the things that make cakes and cookies and soda pop taste so delicious, but are not the most beneficial for your health. Okay. What you should ingest more of will be the most complicated polysaccharides. This should be the principal polysaccharide, or this should be the principal carbohydrate in our diet, I should say. One single polysaccharide can contain hundreds of monosaccharides, just like a brick house could contain hundreds of bricks. The most important and principal one in our body is glycogen. This is what glycogen looks like here on the next page. You can see it's kind of stringy looking figure. All these little purple boxes or oval shaped structures is a glucose. So if you were to go through and count them up, probably 30, 40, 50 glucose molecules. So what glycogen is just a whole bunch of glucose molecules bonded together. Okay, so it's just a storage form of glucose in the body. And we do this that way. If we need energy, we can take those glycogens and just break off the glucose from them individually, and then each glucose can be used to make ATP. Okay. What's nice is we store glycogen in our liver, but even more importantly, even more conveniently, we store glycogen in our skeletal muscles, which are a place where we use a lot of energy. Okay. Because that's where we, do the movement of our body, right? And that muscle contraction process requires energy that we get from glucose in the form of ATP. And we'll talk more about that later in the semester. As far as other polysaccharides, there's 3 other forms. Really, the most important one I want to talk about is starch. Starch and glycogen are essentially the same thing, except the difference is glycogen is how we store our glucose. Starch is how glucose is stored in plants. Okay. You eat potatoes, you're going to find that there's a lot of starch in them. So will pasta dishes. Okay. This should be the major carbohydrate in your diet. Okay. Cellulose is a structural polysaccharide that helps form the cell wall of a plant and their cells. As far as us, though, we can't digest it. It's indigestible, and therefore we can't break it down and get any energy from it. Okay, so it really doesn't do anything for our body that way. It does, however, aid in the movement of feces through the intestines, so it kind of speeds things up, which is very helpful for our health. It is going to help reduce the risk for things like rectal cancer, colon cancer. We commonly call things that have lots and lots of cellulose fiber. Okay? And I'm sure you've all heard of, Fiber One bars and that kind of stuff. And if you have eaten one of those and your body wasn't used to it, you might have been making a mad dash for the bathroom, okay? Because of that increased movement of the feces through the intestines. A great example of this, we live in Kansas, grow a lot of corn here. Corn, for lack of a better way to say it, you eat it and it comes out the exact same way. It doesn't look like anything's changed to it. And that's really because we can't do anything with it to digest it. Last one there, chitin, however you want to pronounce that, is a polysaccharide that fungi have, insects have, but we don't eat a whole lot of that stuff, so it's really something I'm not going to talk about. All right, so that's carbohydrates. 2.6 here table lists off the monos, the dis, and the polys. We talked about each of these different categories, although I didn't get into great detail about the different forms of monos. We will talk later in the lecture about DNA. Deoxyribose is a monosaccharide that helps to form DNA. Ribose, we'll talk about that, how it helps to form RNA. But I'm not going to ask you to directly pull anything from the table. It's included because it's in your book. All right, moving on, then we will now talk about lipids, or as we commonly call them, fats. Lipids can be described as water insoluble organic molecules and are generally nonpolar, making them hydrophobic. So we've got kind of three major things I want to talk about in this first sentence. Water insoluble means that it will not dissolve in water. Okay. So earlier today we talked about like salt dissolves in water. Well, fats do not. Okay, and that's because they are non-polar, or if you want to kind of describe them as having no charge, it's kind of another way you could easily describe it. Probably not the perfect way to say what non-polar means, but for our class simplicity's sake, we'll just say non-polar means no charge. And that word hydrophobic, if broken down, we talked about this in anatomy the other day. Hydro means water, phobic means fearing. Okay, so in other words, fats are water fearing. They don't like water, and that relates to this water insoluble phrase. And that relationship between water and fat is not what you see with everything else. Okay, like salt would be water soluble, and that's because salt is hydrophilic. Okay. Lipids typically make up about 18 to 25% of body mass in a lean adult. Key phrase there is a lean adult. Many people have way more than 18 to 25%. They're composed largely of carbon, hydrogen, and oxygen. However, the amount of oxygen is very low compared to the carbon and hydrogen. Okay. Now, fat, people just think right away it's not a good thing, but naturally we should absolutely have some fat, and that's because it does do a lot of important things for us. Okay. We do have an limited ability to store fat, so our body can take advantage of that to stockpile energy in case we need it. And the reason is because 1 gram of fat actually can store twice as much energy as a gram of polysaccharide. Talk a little bit more about that here in a second. It's a more compact fuel reservoir than carbohydrate. That really relates to that first bullet there. It cushions certain vital organs like your kidneys. There's what we call a fat capsule around each kidney to basically cradle it. In the layer just deep to our skin, we'll find a lot of fat to help insulate, not like blubber that you find in whales, but it does insulate against heat loss. It helps form the plasma or cell membranes of our cells. Some of our hormones are a fat, and fats can help with various forms of digestion. Now, looking at the 6 different categories here, we're going to talk about each one individually. Fats are unique in that there's no individual monomer that is used to build them all. Okay. So in other words, there's no like unifying brick that's used to make everything else. Okay. There is what we call a group called fatty acids. We use fatty acids to make the next 2 categories, triglycerides and phospholipids. But fatty acids have nothing to do with steroids, nor do they have anything to do with the other 2 types. Okay, so fatty acids are as close to the building blocks for fats as it gets. So let's talk about what a fatty acid is. Fatty acids are the simplest of our lipids. And as I've just already said, they're used to synthesize triglycerides and phospholipids, the next 2 fats we'll talk about. 2 forms of fatty acids exist, saturated and unsaturated. We've got an example illustrated here for both. Okay, look at the number of hydrogens and carbons that you see in both these 2 examples. You see a little bit of oxygen in both, but not very much. Okay, the shape is different between the 2. A saturated fatty acid is basically just straight chain of hydrogen and carbons. And the reason it is straight is because from carbon to carbon to carbon to carbon to carbon, you never find a double bond. It's always a single bond. They always use one line for a single. Whereas here where it looks like the equal sign, that means you've got a double bond. Okay. So we say in a saturated fatty acid that all available bonds of the hydrocarbon chain are filled with hydrogen atoms. Whereas here in a unsaturated fatty acid, we have one, in some cases you'll find more than one double bond, which bends it, puts a kink in it. This is what we call a monounsaturated. Once again, there's that prefix mono, so there's one double bond. There's also what you can call polyunsaturated, where there's multiple double bonds. Okay. So a saturated fatty acid, no double bonds, and then an unsaturated has at least one. Does that make sense? Now, if you look on a food label, for example, you will probably find a little segment there where it talks about percent of saturated fats, percent of unsaturated fats. Okay, you might know which of the 2 is worse for your body. It is saturated, and that relates to what we'll talk about here. Saturated fats are really hard on your heart. Okay, the most plentiful lipids, both in our diet and in our body, are the category called a triglyceride. Okay, there are kind of 2 major components used to build a triglyceride: a glycerol molecule, which is a 3-carbon molecule that forms its backbone, and then 3 of these things that we just looked at on the previous page, which we've got a picture of a triglyceride here on the next page. Okay, so looking at this figure, and I'll kind of bounce back and forth between these 2 slides. I really like this illustration because it simplifies things. We've got in blue here our 1, 2, 3 carbon glycerol. There's the backbone, and then attached to it are 3 of these long hydrocarbon chains, each of which is a fatty acid. Okay, so we've got 1 fatty acid, 2 fatty acids, 3 fatty acids, which now I bet you can figure out what we call them: triglycerides. Okay. We either find triglycerides at room temperature in a liquid or a solid state, okay, in a hard or fluid state. Okay. A fat is a triglyceride that's solid at room temperature, and that's due to the presence of those saturated, not healthy fatty acids. Okay. Whereas an oil is just a liquid triglyceride and it's liquid at room temperature because of the presence of those unsaturated fatty acids. That kink, that bend, that is going to occur in a unsaturated fatty acid is going to prevent it from solidifying at room temperature. Okay. This is why anymore you rarely see people cooking with like Crisco, okay, and lard. Okay. You know, my grandma, we got some of her recipes after she passed away. Crisco was in like everything. Okay. And a lot of times, you know, husbands died at age 50 because of all the heart attacks that were happening, right? Okay. Now instead we use oil, safflower oil, canola oil, olive oil. It's a lot better for your heart to cook with oils than to, you know, really grease things up with Crisco. Okay. It does probably taste a little bit better. We say that triglycerides are the body's most highly concentrated form of chemical energy. This means that they have more calories than fat, or excuse me, than carbs or proteins. In fact, more than twice as many per gram as you'd find in carbs or protein. Okay. There are 9 calories per gram in fat in a triglyceride, whereas in a protein or a carbohydrate, it's only 4 calories per gram. Now, we all know what a calorie is, right? You see it on a food label or on the back of a Dr. Pepper bottle. If I ask you though, what does a calorie really mean? It's hard to explain a lot of times for students, okay? A calorie is basically just telling you how much energy is in that food or liquid. The more calories, the more energy it provides for our body, right? Okay. Well, if you have the same amount of carbohydrate, like let's say you have 10 grams of a carbohydrate, 10 grams of a fat, you get way more energy from the fat, and that's why if something's really fatty, it's going to be higher in calories. Does that make sense? So per gram, it's 9 calories for fats, only 4 for carbs and proteins. Okay. I've already mentioned this today, but humans unfortunately have an unlimited capacity to store triglycerides in adipose tissue. So even if we don't ingest a fat, our body, like in the case of excess dietary carbs, proteins, fats, and oils, we can convert those to triglycerides to be stored in adipose tissue. So if all you ate was cookies and cake all of your life, which is really just sugar, you could get fat because your body can convert it. We'll talk more about that later on the semester. Okay, very similar looking to triglycerides and very similarly structured to triglycerides are the next fats, what we call phospholipids. Okay, and they are similar because they have a glycerol backbone, just like our triglycerides do. They've got fatty acids, except now they've only got 2, and you can see they are shown here in gray. Here in this portion of our figure right there, as I circle it, is the glycerol. That third carbon that you see doesn't have a fatty acid tail coming off of it. Instead, we have some stuff going in the opposite direction, including a phosphate group, and then all this other stuff. I won't really get into that. But this creates a really, really unique type of fat. That's what we call amphipathic. Okay. Amphipathic means that it's got a polar head and 2 non-polar tails. Now going back to this slide, we said that fats are generally non-polar, making them hydrophobic, which we said means that they don't like water. They're afraid of water. Okay. Well, the tails of a phospholipid are just like any other fat in that they're afraid of water. Okay. Like if you put oil and water together, they don't mix. Okay. That's how these tails operate. But the head is not hydrophobic, it's hydrophilic. It likes and is attracted to water. Okay. And we talked about this in anatomy. I don't know if several of you are in that class, so this is kind of a double dose, but I'm talking about it, hopefully terms that make a lot more sense now. Okay. Well, if you have a whole bunch of these guys, which our body does have a whole bunch of them, when exposed to a liquid environment, they are going to create a really cool structure called a bilayer, which is 2 layers of these phospholipids organized where the heads are aligned and pointing in opposite directions of one another, where the tails are in the center pointing towards one another, away from the water, whereas the heads are pointing towards where the water is, inside and outside the cell. We're going to talk a lot more about phospholipids in our next lecture and their role in forming what we call the plasma or cell membrane, the outer boundary of a cell. All right, looking at the 4 illustrations at the bottom of this page, we see a big change in structure. This type of fat, what we call steroids, made up of a 4-ringed hydrocarbon skeleton. These A, B, C, D rings that you see here are those 4 rings, okay, making them structurally different from other lipids. So instead of being long chains, they're more ringed in structure. Okay, most common steroid in animal cells, we are an animal, would be letter A here, cholesterol. Okay, in fact, we use that cholesterol molecule as the starting point for synthesizing any of the other steroids in the body, including a very important hormone in women, estrogen, testosterone, very important male sex hormone, cortisol, a hormone that helps us resist stress. Okay, so there are many steroid hormones in the body, but steroid hormones are not the only form of steroids. There's also bile salts that help us digest dietary fats, vitamin D, among other important things we'll kind of touch on at various points in the semester. Okay, last thing I want to talk about for fats include eicosanoids, fat soluble vitamins, and then lipoproteins. So it's just kind of whatever's left here on this slide for fats. Eicosanoids are fats or lipids composed of a 20-carbon unsaturated fatty acid called arachidonic acid. There's 3 principal subclasses of eicosanoids: prostaglandins, thromboxanes, and leukotrienes. Prostaglandins play a role in regulating the effect of several hormones. Thromboxanes constrict blood vessels and promote platelet activation. That's something we do like when we have a blood clot or when we're trying to form a blood clot. Leukotrienes participate in allergic and inflammatory responses. Okay. These guys are kind of like mediator molecules. They might not produce a drastic effect to the body, but they might help something else do a more drastic thing. So we don't talk a lot about these. They're very important to us, but there's just other things that we cover a little bit more in detail than those 3 different eicosanoids. And then finally, other lipids include our fat soluble vitamins. That includes the beta carotenes, which beta carotenes produce the yellow orange pigment in the egg yolks, carrots, and tomatoes, which we convert to vitamin A, vitamins D, E, and K. So A, D, E, and K, those are the 4 vitamin fat soluble vitamins. And then the lipoprotein, we don't really talk much about any of this stuff. The lipoproteins are kind of what exactly it sounds like, a lipid protein kind of hybrid. It's when a lipid joins with a hydrophilic protein molecule, which is important. This is what we do for making them more soluble in our blood plasma, with proteins on the outside so they can get carried in our bloodstream. If you've ever heard of HDLs and LDLs, anybody, does that ring a bell to anybody? If somebody ever goes to the doctor and they have like a blood test done and they're checking like cholesterol levels and that kind of stuff. They're also going to check the levels of lipoproteins. The prefix or the abbreviation HDL that I just said is short for high density lipoproteins. LDL is short for low density lipoproteins. So there's measuring a form of fat that you'll find in your blood in the case of those two things. If you take any like exercise or nutrition related classes, you probably learn a lot about those. I say probably you would, but we don't really get into them too much in this class. All right, so that's our fats of the body. Once again, just another table, just like I gave you a couple slides ago when we had carbohydrates, breaking them down individually. Not some I'm gonna ask you to directly study because we've already kind of covered it all. That brings us to our third of the four categories, and that is proteins. Proteins are very large molecules making up about 12 to 18% of a normal lean adult body. And I love the second sentence there on this slide, stating that they're instrumental in almost everything cells do. Okay. I love the very last sentence on this page as well. Chances are if something is getting done in your body, it's being performed by a protein. And so if something's happening, there's a good chance it's protein doing it. Not always, but when in doubt, you could guess protein and you probably would be correct. Okay. Proteins are linear polymers. They're long chains of what we call amino acids. Here's what an amino acid looks like. They're bonded together by what we call peptide bonds. Okay. Proteins are much more complex, not only in structure, but also in function than carbs or lipids. And there are 20 different amino acids. Each is made up of kind of four major things. Okay. At the center is a carbon. On one side, you'll find an amino group, NH2. On the other side, you'll find a carboxyl group. Then you're going to find a singular hydrogen atom bonded to it. And then, now this is kind of misleading. It's not R as in that's an atom. That would be what we call a side chain or R group, which is going to be a collection of atoms. They just kind of simplified the illustration here. Okay. There are 20 different amino acids. Okay. Like methionine, tryptophan, leucine, isoleucine, so on and so on and so on. Okay. You take two of those, you bond them together, you form what's called a dipeptide. A third would form what's called a tripeptide. If it's between 4 and 9, we would just call it a peptide, and 10 or more, we'd call it a polypeptide. Okay. Think of amino acids like letters in the English alphabet. Okay. So how many letters are there in our alphabet? 26. Okay. And now here's a question that you're not going to be able to answer because it's kind of impossible. How many words are in the English language? Right. It's not common. How do you form a word? A specific sequence and number of letters, right? Well, how do you form a protein? A specific number and sequence of amino acids. Okay. If you move letters in a word, does the word change? Absolutely. If you move amino acids in a protein, it changes. Okay. So because each variation, the number or sequence of amino acids can produce a different protein, a great variety of proteins is possible. In other words, proteins are like words. There's a heck of a lot that can be formed because you just put things together differently, just like you do with letters to make different words. And what's amazing is each protein has a unique function. And as I've already mentioned, chances are if something's getting done in your body, it's performed by a protein. And that's demonstrated here with the table 2.8, listing off the 6 different functional types of proteins. This is an actual table I do want you to know. So put a star in it, highlight it, whatever you need to do. There might be, or probably will be, questions related to this on the exam. Proteins throughout our body can provide structure. In class this morning in anatomy, we talked about collagen. We also will talk more in our next lecture about keratin in anatomy. That's a very important protein, not only in our skin, but also hair and our fingernails. Okay. There are proteins that are regulatory, functioning as hormones, like the hormone insulin that everybody hears about when people discuss their diabetes. Okay. Insulin regulates your blood sugar. Insulin is a protein that regulates. Okay. Two major proteins, myosin and actin, are contractile. They allow our muscles to shorten, producing movement. And we'll talk about myosin and actin quite a bit when we get to our muscle lecture here in about a month. Okay. Our proteins can function for immunity, like our antibodies that are produced to fight off things like the coronavirus. Okay, so they provide protection against foreign substances and invading pathogens. We have proteins like hemoglobin that transport things throughout the body. Hemoglobin is a very complex protein that transports our oxygen and a little bit of carbon dioxide in our blood. And one of the most important, and we'll talk a little bit more about this here in the next couple of slides, are how some proteins can be catalytic, meaning that they act as enzymes to help speed up chemical reactions, like the salivary amylase one that's listed there that helps us digest our carbohydrates that we've eaten. So make sure you know the table and what these different categories are. All right, the shape of a protein, the structure of a protein is very important. Each protein can have its own unique structure, and this variation in structure and shape is going to be directly related to their diverse function. And I've told you guys this before, but I love analogies. And I like to think of the importance of protein structure like the slinky. Okay, everyone knows what that slinky is, right? Okay, and we've even got a type of protein called an alpha helix that kind of looks like a slinky. Okay, you don't know what a slinky is? It looks like a metal circular structure that walks down the stairs. Okay, everybody has them as a kid, and they last about a week before they get all tangled up and no longer work, right? Okay, well, proteins are like a slinky in that they can lose their shape, and this change in shape can be very detrimental to their function. Okay, what can change the shape of a protein? Its environment surrounding it, specifically the temperature and the pH of the fluid, which can cause it to unravel and lose its shape. So like when a slinky gets a knot in it, that's what we call denaturation, or denaturation. A protein becomes denatured, it loses its shape, and as it says down here at the bottom, is no longer functional. Okay, and what did we just learn? If something's getting done in your body, chances are it's being performed by a protein. Okay, this should help you understand why it's so important to regulate your body temperature so precisely. Okay, we like our body temperature to be 98.6 degrees Fahrenheit or 37 degrees Celsius. We can go above or below that just a few degrees, but if your body temperature gets up to like 107, 108, you're not going to be in good shape, nor is it going to be good if your body temperature drops to like 92 degrees. Okay, those are situations where proteins start to unravel and then they stop functioning. And like I said, if something's happening, good chance of proteins doing it. And if your whole body starts to get too hot or too cold, all those proteins start shutting down. Your whole body starts to shut down. Okay, does that make sense? Okay, and that explains also what we talked earlier today about how like the pH in your stomach is very different than the pH of your mouth versus the pH of your blood, and why it's important to maintain the pH of your blood between 7.35 and 7.45. Okay. There's kind of 5 major categories for protein structure. The simplest is a primary structure with each of these little boxes, just a amino acid, bonded, 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 bonded. Then you get into alpha helices and beta pleated sheets. Those would be at the secondary structure. Then tertiary when you got a collection of both of those, and then most complicated would be quaternary, and you've got several tertiaries. I'm not really going to get into it. Just know that proteins can come in all sorts of different sizes, shapes, which affects their functions, and shape is a very important thing, as I've already mentioned. Now, one of the, was it 6 or 7 different types of proteins as far as function goes? Was the very last one catalytic? So let's talk about what that word catalytic or catalyst means. Enzymes are proteins that are considered biological catalysts. A catalyst is a chemical that increases the rate of a reaction, is not itself changed at the end of the reaction, and doesn't change the nature of the reaction or the final result. So in other words, all it does is speeds things up, okay? And how it all works is it affects what we call the activation energy, which is the energy or the amount of energy required for a reaction to proceed, okay? So in other words, it's how you go from the beginning of a reaction to the end of a reaction, okay? Here comes another analogy, okay? They're not quite as popular, I'd say, now as they were maybe 10 years ago, but those like obstacle course races, Tough Mudders and whatnot, where you've got, mud pits and, pools that you have to swim through and then walls that you have to climb over. Let's pretend that we're all taking part in a Tough Mudder event and we're coming up to the very last obstacle, and on the other side of the obstacle is the finish line. Okay, the last obstacle is a wall that we have to climb over. Okay, and we have 2 choices. We can climb a 10-foot wall, or we can climb a 5-foot wall. Okay, obviously, one's taller than the other, and it's going to take longer to climb the taller wall, right? Okay, well, we're going to cross the finish line no matter what. But if we choose the 5-foot wall, we're going to get across that finish line faster. Okay, well, an enzyme is going to take you from the 10-foot wall, shown here in red without the catalyst, to the blue line here, which would be like the 5-foot wall. We've lowered the barrier to get from point A to point B, so we get there faster with a catalyst, with an enzyme. The end result is still the same, crossing the finish line. It's just, once again, we've increased the rate of reaction. Okay, I would highlight this last bullet here. Catalysts speed up reactions at a lower temp by lowering the activation energy required. So they lower the obstacle to get from the beginning to the end of the reaction. That makes sense? All right, we'll stop here because we are right up against time. We'll finish up talking a little bit more about enzymes on Thursday. And then we'll move on into our last lecture on cells at that point.